Antibody structure, contents, antibody units, affinity, antibody valence and avidity. Molecular components. Antibodies are glycoproteins that bind antigens with high specificity and affinity. They hold on tightly. They are molecules originally identified in the serum, which are also referred to as immunoglobulins, a term often used interchangeably with antibodies. In humans, there are 5 chemically and physically distinct classes of antibodies: IgG, IgA, IgM, IgD, and IgE. Antibody units. All antibodies have the same basic polypeptide chain unit, 2 light or L chains and 2 heavy or H chains, as shown in the figure. In this basic unit, one L chain is bound by a disulfide bridge and non-covalent interactions to one H chain. Similarly, 2 H chains are bound together by covalent disulfide bridges as well as by non-covalent hydrophilic and hydrophobic interactions. There are 5 different kinds of H chains referred to as mu, delta, gamma, epsilon, and alpha chains, which determine the class of antibody IgM, IgD, IgE, IgG, and IgA respectively. There are also 2 kinds of L chains, kappa and lambda, each with a molecular weight of 23 kilodalton. There are also 2 different kinds of L chains, kappa and lambda, each with a molecular weight of 23 kilodalton. Each antibody unit can have only kappa or lambda L chains, but not both. The properties of the different antibody classes are shown in table 1. Properties of human immunoglobulins. The physical properties: molecular weight in kilodalton, IgG 150, IgA 170 to 420, IgM 900, IgD 180, and IgE 190. H chain molecular weight kilodalton, IgG 50 to 55, IgA 62, IgM 65, IgD 70, and IgE 75. Physiological properties: normal adult serum milligram per milliliter, IgG 8 to 16, IgA 1.4 to 4, IgM 0.4 to 2, IgD 0.03, and IgE is in nanograms. Half-life in days: IgG 23, IgA 6, IgM 5, IgD 3, and IgE less than 3. Biological properties: complement fixing capacity is present in IgG, absent in IgA, and present in IgM, IgD, and IgE doesn't have them. Then we can also see their role in anaphylactic and placental transport to fetus. Both H and L chains have intrachain disulfide bridges every 90 amino acid residues, which create polypeptide loops, domains of 120, 110 amino acids. These domains are referred to as VH, VL, CH1, CH2, and so forth, and have particular functional properties, example VH and VL together to form the binding site for antigen. This type of structure is characteristic of many other molecules, which are thus said to belong to the immunoglobulin gene superfamily. The N-terminal half of the H chain and all of the L chain together make up what is called the Fab fragment in the figure that contains the antigen binding site. The actual binding site of the antibody is composed of N-terminal quarter of H chain combined with N-terminal half of L chain. The amino acid sequences of these regions differ from one antibody to another and thus called variable or V regions, and contain amino acid residues involved in binding an antigenic determinant. Most of the antibody molecule The C-terminal three-quarters of the S chain and the C-terminal half of the L chain are constant C, that is, have the same amino acid sequences as all other antibodies of the same class and subclasses. The C region did not bind antigen but rather determine the biological properties of the molecule and thus the fate of antigen bound by antigen binding site. In particular, the C-terminal half of the S chain, the Fc region or fragment crystallized, serves another other function, that is combines with complement, is cytophilic, or binds to receptors in certain types of cells such as macrophages and so forth. Carbohydrates are also present in antibodies, primarily on the Fc portion of the H chains. Affinity. Different antibodies molecules produced in response to a particular antigenic determinant may vary considerably in the tightness of binding to that determinant, that is, in their affinity for the antigenic determinant. The higher the binding constant, the less likely the antibody is to dissociate from the antigen. Clearly, the affinity of an antibody population is critical when the antigen is a toxin or virus and must be neutralized by rapid and firm combination with antibody. Antibodies formed soon after the injection of an antigen are generally of lower affinity for that antigen, whereas antibodies produced later have dramatically greater affinities, association constants 1,000 times higher. Antibody valence and avidity. The valence of an antibody is the maximum number of antigenic determinants with which it can react. For example, IgG antibodies contain two Fab regions and can bind two molecules of same antigen or two identical sites on the same particle and thus have a valence of 2. Valence is important for the binding affinity, as having two or more binding sites for an antigen can dramatically increase the tightness of binding of the antibody to antigens on a bacteria or virus. This combination effect, avidity, results from synergy of the binding strengths of each binding site. Avidity is the firmness of association between the multi-determinant antigen and the antibodies produced against it. Determining the avidity of an antibody population is difficult since it involves evaluating some function of the group interactions of a large number of antibodies with a large number of antigenic determinants. Even so, the importance of avidity can be demonstrated both mathematically and biologically. For example, as a result of working together, being on the same molecule, two IgG binding sites are 10 to 100-fold more effective than neutralizing a virus than two unassociated binding sites, and if antibody has more binding sites, as in the case of IgM, section D2, it may be 1,000,000 times more effective. This can be visualized by considering antibodies with one or two binding sites for a particular antigenic determinant on a microorganism. The antibody with one site can bind to but can also dissociate from a determinant of an organism. When it comes off, it can diffuse away. However, the antibody with two sites can bind two identical determinants on the organism. Each organism has many copies of each protein or carbohydrate. If one binding site dissociates, the other is probably still attached and permits the first site to reform its association with the organism. It therefore follows that the larger the number of binding sites per antibody molecule, the larger the number of bonds formed within an organism, and the less likely it will be to dissociate. Thus, an antibody with a poor intrinsic affinity for an antigenic determinant can, as a result of large number of combining sites per molecule, be extremely effective in neutralizing a virus or complexing with a microorganism.